So it's my pleasure to share with you our current results in this research project where we try to investigate the potential of Impella support for recovery of chronic heart failure. And so we all know that implantable LVADs can create reverse remodeling in chronic heart failure, and in some cases even improved function and recovery. And so the goal of this research project is to find out if unloading by Impella can lead to the same reverse remodeling in chronic heart failure, to describe the functional, myocardial function, but also cellular and subcellular effects, and to gain insight in a possible optimized mode of unloading, which is possible with the sensorized Impella device. So in order to do so, we used our ischemic chronic heart failure model. And this chronic heart failure model is made by a combination of a permanent ligation of the diagonal branches together with an ischemia reperfusion injury by temporarily occluding the LAD in its middle section. And that creates then an ischemic cardiomyopathy with partially scar, partially borderline zone, and partially remote zone, very similar to a clinical ischemic cardiomyopathy. So we learned from this model that it leads to an increase in end diastolic volume, an increase in end systolic volume, a reduction in the ejection fraction, increase of the sphericity index, and hypertrophy of the myocytes with left ventricular mass that increases. The stroke volume remains the same, which is the reason why the heart remodels in the first place. So we used this model then to randomize animals between being Impella supported versus control animals. So the model by itself has quite a lot of mortality together with unpredictable effects, and we decided to include only animals in the study that have a dilated cardiomyopathy with an ejection fraction less than 40%. So the infarcts were made in 40 animals. We included 16 in the randomized study after 8 weeks, after it was confirmed that they had a dilated cardiomyopathy with a reduced ejection fraction, and then half of them went to sham control, and half of them were supported with the Impella 5.5, running at maximal unloading, just preventing suction, and this for 3 months. And then the animals were terminated. 2 animals were for technical reasons excluded, so we had 7 animals in each group to compare. We did MRIs, hemodynamic measurements, biopsies, blood samples at the beginning, just before the randomization, and at the end of the study. So we hope to find results, first of all, functional, structural changes in the myocardium and function of the ventricle, on the level of fibrosis and extracellular matrix, based on the biopsies taken and genetic expression of the myocyte itself, as well as in blood biomarkers that might give a hint which animals will do better than others. Today, I'm going to present only the result of the functional data, because these are the data that we have already. So here you see the data of the MRI measurements in these animals, in red for the ones that have been supported with the Impella device, and in black for the control animals. You see the individual evolution of end diastolic volume and systolic volume, stroke volume, ejection fraction, cardiac output, and sphericity index at the different time points. Of course, they were only supported between 8 weeks when they were dilated, up to 5 months at the end of the experiment. You can tell that there is some variability, that there seems to be an improvement in the volumes and diastolic and end systolic volume for the ones that were supported, and that some animals had an improved ejection fraction, the ones that were supported, but not all. So there is variability, and clearly the animals that were dilated most showed the best potential in reversed remodeling. So looking at it by a two-way ANOVA and comparing the control group with the supported group, there is a significant reduction by Impella support of the end diastolic volume and end systolic volume. There is no significant group effect on the stroke volume, sphericity index, cardiac output, or ejection fraction. This observation that the ejection fraction is not significantly changed in a group effect sort of reflects to the clinical reality that we often see with LVADs also, reverse remodeling, but not in all patients, functional recovery. But of course, this is an ischemic cardiomyopathy model with a big scar, and the scar size did not change from 8 weeks to 5 months support. So we did a deeper dive in the analysis of the remote zone, the healthy part of the heart. And if you look there at the MRI analysis, we do see a regional function improvement in all the supported animals, which is significant in all measures that we could make. The regional ejection fraction of the remote area, this is like only the slice that is analyzed where there is no scar tissue involved, or the remote wall thickening as measured by MRI, or the remote endocardial area change, which is a very similar measure as the regional ejection fraction, all were significantly improved for the supported animals as compared to the non-supported animals. Secondly, the remote wall volume measured by MRI was significantly reduced in the supported animals, and so this hints to less muscular hypertrophy. And this is also confirmed by the histology that we have already on the biopsies taken in the remote zone, where there is a significant reduction in the mean myocytes. So there is in the mean myocyte site. So in other words, the hypertrophy is reduced in the animals that were supported. Now, combining the pressure data that we have and the serial volume data that we have, we are able also to depict pressure volume loops in the same animal over time. As you can see here in a control animal at baseline, at 8 weeks, and then at the end of the experiments at 5 months when there is further dilation and remodeling towards heart failure. And as you can see here in a supported animal, there is reverse remodeling in the green curve, as you can see, after 5 months as compared to the status at 8 weeks after the heart was remodeled in the first place. So our analysis will progress. We are awaiting the results of the plasma samples that have been sent to New Zealand for analysis. We are further looking into the biopsies in terms of the extracellular matrix and the genetic expression on myocyte level. We are further analyzing the PV loops and the pressure data that are actually derived from the impeller pressure sensor itself in order to find out how we can further determine which animals are doing the best and also to determine when they are recovering. But based on the data that we have so far, I think we can conclude that impeller support reversely remodels the chronic failing heart in a significant reduction in end diastolic and end systolic volume and a reduction in hypertrophy, a finding that we had to exactly the same extent with full implantable VATs in this same animal model. The most dilated hearts were the one that remodeled most, and impeller support improved as well regional contractility in the healthy zone in this model of ischemic cardiomyopathy with scar, and this gives also some hope for the clinical scenario for non-ischemic dilated cardiomyopathies. I thank you for your attention.